on virology, I would certainly go heavier on this stuff. But this stuff will be on there as well. So the student who knows more will get a higher grade if I do this right. Okay. Promise to return to the paper since it's the most interesting thing I've done this semester. But I'm going to go a little out of order and go chapter 24, and then back to the papillomavirus paper. Chapter 24 is pretty short. Virologists want to be able to kill viruses. So the chapter is, I think it's called Survival of Infectivity. That's a proxy for alive, but of course there's debate about whether viruses are living or not. So being infective is the viral analogous state to being alive. So viruses die over time. And they are small and they're simple, except where they're not. I will ask you about the size of viruses on the final. There's the panel. So, why do they die over time? Because they are susceptible to variations in temperature, just like you and I. Radiation. Here we're largely talking about ultraviolet radiation that damages nucleic acids, but of course you can also bombard them with beta particles and whatnot in terms of sterilization. And then chemicals and environmental stressors. E.g. pH. We don't like to get hot, viruses don't either. We don't respond well to radiation, and we don't like very acid or basic environments. The same is true of viruses. I forgot my spreadsheet. So, here's a virus dying over time. This would be a loss of infectivity over time as a result of a one-state process. So if you say move a virus to a higher temperature, decay, this is percent surviving infectivity. If you start to sample all live viruses and just wait over time in a given condition, it will decay into non-infectious. I wanted to illustrate at least the mathematics of this, and I put up a big spreadsheet. I forgot to bring it. Sorry, you guys did want me to slow down, so I'm going to recreate my spreadsheet and slow down at the same time.
Showing off a little too much leg there. That's sexy.
I'm just going to generate some virus infectivity survival data. Okay, this is the rough cut of it. So I've generated some just infectivity or percent infectivity data based upon a simulation, and you can see that linear relationship over time on a log scale, so 110:1, it is actually a result of an exponential decay. So you've seen this hopefully throughout your undergraduate career in things like decay of radioactive isotopes. So there's a time function that just made up units of time, maybe they're minutes, maybe they're seconds, that a decay, so from 100% over time virus infectivity drops. That drop is dependent upon a rate constant, which I've just made up there, 0.1. And so if you look at the Excel version of my calculation, I plugged in an exponential decay, which is where it starts. I just call that 100 because my scale is percent. And then the decay is an exponential function of time, so that defines this cell as the time component. And then it's e to the minus k times t times. So this is just a one-phase exponential decay. 0.1 is very, very fast, so it's a per whatever time unit this is, let's call it seconds. So it's a per second rate constant. And then you see a very fast decay. So you would describe a viral decay based upon that rate constant. You know the rate constant. You can, of course, describe a virus at a 10 times slower rate constant, 100 times slower rate constant, very slow rate constant. So a more stable virus will have a lower rate constant of loss of infectivity. Let's call it K loss. And then you can make yourself a spreadsheet that shows that. But note that as I change the numbers here, the log function is always linear. Going back to this, your text tries to show a two-state process as a sort of a step function where we're declining fast and declining slow. In reality, a multi-phase decay is going to be the sum of two or more exponential decays. So if you have a virus, you would guess heating it up might cause an increase in the rate of loss of infectivity, but if you heat it up and have it at, say, an acidic pH, that would be two factors in its decay. You would expect to see something more akin to this, and in reality it would be a two-phase curve, which I did not generate. Well, that's not much of a curve. So this is one state, but you could see a drop, and then the slower phase would have us, would be like this plus this, so it would be like that, and you could see the two phases. Do note, nature of an exponential decay, of course, is an asymptotic approach to zero, but technically speaking, it's never zero. My lowest number here is times 10 to the minus 26. That's pretty darn close to zero. But this is a problem we'll see in killing all viruses in vaccines. Technically speaking, never kill them all, at least as a single exponential. So unsurprisingly, most viruses are going to be less stable at higher temperatures, and you see this is a phage, very stable at 55, so that has a very low rate constant, but as you heat it up, you get different rates of decay as a function of temperature.
Can you get nature viruses in
Yes, that is one of the primary reasons you autoclave things, is to kill the viruses. Or the yeast, or even the pneumatic itself. Radiation, ultraviolet radiation causes thymine dimers, so a cross-link between the aromatic bases in the nucleotide, which results when it resolves in a mutation. Remember, viruses don't have proofreading, so they tend to be more susceptible to ultraviolet radiation than we are. One reason why food is bombarded with, well, not ultraviolet radiation, but food is bombarded with radiation to kill microbes and viruses. Here's a virus that loses infectivity much faster the lower the pH, so it doesn't like acidic environments. Why would an acid environment inactivate the virus? Right. Hydrolysis. Yes, same thing. Just the way you and I would react to acid, the more protons is faster hydrolysis reactions. So, you have lots of reasons to kill viruses. One, we want to make vaccines. We'll find out in the next chapter. Many vaccines are made with inactivated viruses. We want to eliminate them from medical products. We want to eliminate them from food. And then research tools. And all of this can be done in an autoclave by subjecting them to high temperature and pressure that keeps the water from boiling in an autoclave. But one of the reasons why you autoclave things is to get rid of viruses.
Does that say research tool?
Research tools, yes.
Okay.
So your pipette tips are a research tool. You generally don't want viruses in for lots of reasons. So those chemical factors, pH we've already mentioned, hypochlorite, which is of course bleach, kills them by oxidizing, which inactivates a number of things. Aldehydes create chemical crosslinks between viral biomolecules, thus inactivating them that way. And then lastly, at least I mentioned in the textbook, detergents. You can dissolve lipid membranes and unfold proteins with detergents. So that's why, or one reason why detergents and bleach are in cleaning products is they kill viruses. Travis.
What was it you said about the aldehydes?
They create cross-links. So formaldehyde, for example, or glutaraldehyde is the other one mentioned in your text. Cross-link proteins. Cross-link more than that. But that inactivates them by creating chemical bonds. Other things. And here's another set of decays based upon the parts per million of hypochlorite. And that's it. Oh, one last point. Envelope viruses are generally more susceptible to loss of infectivity over shorter times than naked. Why that might be will be an excellent potential final exam question.
Can you repeat that? Envelope viruses.
Are more susceptible to loss of infectivity in any given, generally speaking, in any given condition. Why might that be? Just gonna leave that hanging there because I think if I can remember it, I'll use that. So, potential final exam question. Anybody got an answer?
Yeah,
That's also true. Naked side. You're getting down the road. Okay, let's revisit my apparently favorite paper in the world. And you, as an aside, I posted a paper earlier for this coming week, and then I started looking at it. Seemed like an interesting topic, it may well be. But it was huge and complicated. I thought, I've done it to them once already. So I picked a 3-page paper that is not freely available, so it's through interlibrary loan. Try to have it for Wednesday. When I get it, I'll post it, but I won't expect you to have read that one closely by class. But it's short, but also interesting. Okay, remember our viruses and cancer chapter where we have HPV causes cervical carcinoma, expresses 2 gene products that are proteins E6 and E7 when translated. E6 binds P53, a tumor suppressor protein. E7 binds PRB or 105, also tumor suppressor protein. Thus repressing the tumor suppression pathways and resulting in cancer. So just to recap, we've got 2 strains of HPV that are known to more frequently cause cervical carcinoma, 16 and 17. We have viral proteins E6 and E7. E6 binds P53, leading to destruction. E7 binds PRB, aka P105 RB. Viral protein E2 is a negative regulator of E6 and E7. So when E2 is present, transcription of E6 and E7 are shut down. So hopefully I can get through this part in about 2 minutes today. Now, figure 1, northern blots are looking at transcript levels of either just the vector alone. So they're going to infect. Sorry, more background information. Remember the HeLa cells from Henrietta Lacks' cervical carcinoma are infected with HPV 18. They're expressing E6 and E7, E being for early, and the tumor suppression pathways, both the P53 and the PRB pathway, are shut down in HeLa cells. Thus, those cells are tumorous and continue proliferating in addition. If you go read the book, you'll see that they're amazingly proliferative. So, the group that published this paper took either a virus that's just empty or express, oh, sorry, this is minus and plus, either are infecting these cells with a virus that will express large amounts of E2, or a mock infection where E2 is not produced. There are 3 different constructs of HeLa cells that have either empty vector or a copy of the E6 or E7 gene from strain 16. In this paper, always on. So when you see 16E6 or 16E7, those are cells that have had a copy of the gene for E6 or the gene for E7 transfected into the cells, not under E2 control. So that from this panel we see if we have HeLa cells not transfected with an E2, they express E6 and E7. And when you infect them with a virus that delivers E2, transcription is shut down. E6 and 7 go away. The RNA is no longer made. Because these are separate clones of each of the strains. So they're looking at everything as a function of 2. So and then these 2 panels are screening for the presence of transcripts of the strain 18, 6 and 7, or the strain 16, 6 and 7. Remember, HeLa cells have strain 18 in them, and they have probes that can differentiate between the 2. So a 16 probe won't bind to the 18. So we see in the 16E6 and 16E7, when we infect with E2, we also shut down transcription just as we do without this constitutively expressed gene. However, that 16 gene, again, heterologously provided, so it's not part of a HeLa cell. We see that 16, whether E2 is there or not, expresses E6 and 7. 16E6 expresses E6 and 16E7 expresses E7 from strain 16, not strain 18. The reason to do this was to tease apart the effects of E6 and E7 on proliferation of the cells. That's what we're doing in the rest of the paper. I think that went much better. This, however, is the stuff. All right, so now we're doing Western blots. We're looking at protein levels and whether or not delivery of E2 shuts down repression of the tumor suppressor pathways. Looking at the presence of proteins. So we've got P53. So remember, E6 shuts down, binds P53, leading to its destruction. P53 activates P21. So P53 activates P21, which shuts down CDK2. So CDK2 cyclin-dependent kinase. CDK2 is active when the cell cycle is moving into S phase. So we have P21, cyclin A. So cyclin A is a protein that's expressed when the cell has a role in moving the cell from G1 to S. So E6 causes the destruction of P53, which doesn't activate P21, which doesn't deactivate cyclin A. So the proliferation caused by, this arrow should go this way, this proliferation marked by A is controlled via P53. So we can see that in the LXSN strain, no E2, no P53, and that's because E6 is binding to P53. If we shut down E6, we get P53 back. P53 and the presence of P21 are also correlated, both clones. Each set of things is identical here. In the case of 16E6, when we provide E2, that E6 from HPV 18 is shut down because it's under control of the promoter that's repressible by E2. But that strain 16 that's always on is still on. P53 is not there because it's being destroyed. So that's why there's an absence here. So that presence of E6, which is what binds P53, is sufficient to destroy or cause the destruction of P53. And it's not correctable because we can't shut off E6 expression with E2 when it's being delivered via another vector. In E7 is always there, but E6 is suppressible by E2, and so we see the same thing as in the non-transfected cell. Now, E7 is going to cause the destruction, or it's going to bind to and inactivate PRB, and thus we're going to lose that suppression pathway. P1 or PRB or P105 is activated by hypophosphorylation. So this is inactive, active. So this is a little bit more phosphorylated than this. I'm sorry, other way around. Phosphorylated and then the hypo is the inactive. So the point being, if it's higher in the gel, it's inactive. If it's lower in the gel, it's active. And so we see in the control strain that expression of E2 represses E7, allows activation of PRB. In the case of E6, we get the wild type because we're delivering E6 but not E7. Over in E7, and this is less clean than the P53, we see a loss of active PRB. So E7 is not suppressible by E2, it continues to suppress PRB.
Hold on, say that last part again. E7 is not suppressed by E2, it continues to suppress PRB.
Yes, but I didn't give that enough qualifiers. Remember, we have two different E7s. We have the E7 from HPV 18 that is under control of E2. Where E2 is a transcription factor, so it's going to bind the DNA and shut down expression. When in the two constructs, 16E6 and 16E7, either E6 or E7 from strain 16 is being provided under a different promoter. So the E2 binding site is not there. So in E6, infection with E2 shuts down E7, but not E6. It shuts down the E6 from HPV 18, but the one from 16 is still there. The converse is true over here. I thought this was such a great paper that I got into it. I was like, oh, shit, it's complicated. I was lucky I didn't get the job. Okay. I'm sorry, I got cyclin A mixed up here. So forget the cyclin A over here. P21 CDK2. So the other E7, it activates PRB, among other things. And we'll see CMYC as well. So looking at downstream of P105 is cyclin A. See that if we activate RB, we lose the cyclin A. Cyclin A is what's pushing us into the S phase. So in E6, it's E7 that's shut down, that stops RB. It's reactivated here in the infected one, but over here E7 not being suppressible leads to persistence of cyclin A and thus driving proliferation of the cells. CMYC is another downstream, or another downstream protein of RB. We lose it when the suppressor pathway comes back on. It's not, again, not as clean, but we have much lower levels of it in the case where E7 is not suppressible. This figure is this is sort of a reality check. I'm sure the authors thought this was just the bomb, submitted the manuscript, and the reviewer said, oh, there's this other protein, P16INK, that might be involved in this. And so I would bet a lot of money that this was added in review to show that P16 is not involved, because it doesn't change. Somebody objected, they did this control, and perhaps they thought of the control ahead of time, but it has all the hallmarks of being something put in response to reviews. So CMYC is an oncogene. It's a downstream. It's a DNA binding protein. It's a transcription factor. So when you activate CMYC, you turn on a bunch of genes. It's a hallmark of proliferation.
So, Joe, I missed like the beginning part. Like how it's going with the figure.
So CMYC is the activity of PRB keeps CMYC from being expressed. So in the case of the proliferation, so let's just take this lane right, or these lanes right here. So this is the normal HeLa cell. Doesn't have either tumor suppressor pathway on. Lots of CMYC. We express with E2. We shut down 6 and 7. So this doesn't have the exogenously added strain 16 proteins. Much lower CMYC levels. That tumor suppressor pathway being on results in a decrease of CMYC levels. Genes that are turned on in the proliferating cell are turned off because they're not being turned on. When E7 is expressed, it inactivates RB, leading to more CMYC, leading to proliferation. That's why the big fatty right there, and it's going there.
Is the separation on the chart of this 16:6/15:7? Was that what they're talking about when they say exclusive expression?
Yes.
That one part right there, okay.
And for simplicity, I'm not going to speak much about that other than these are, so in the, they're teasing it apart here and here, they're putting it back together here essentially, and they're retaining the proliferative state in every condition because they can't turn it off. The right to are controlled. Okay. So, how does cyclin-dependent kinase turn on cell proliferation? It does a number of things, all of them are phosphorylations. It phosphorylates histone protein 1. Remember, histones are what chromatin is packed, or DNA is packaged in chromatin, so DNA is wrapped around the histone proteins. CDK2 phosphorylates histone 1. Phosphohistone 1 is open. So DNA packaged around histones, histone gets phosphorylated, the DNA is essentially unpackaged. So phosphorylated histone 1 is open for business in terms of replication, synthesis, and cell division. Your DNA is packaged up in histones, it's not expressed. And so figure 3 is a pull-down experiment. Jargon, they call it a pull. I don't think they even define it as, they call it a pull down. It's a co-purification experiment. So they purify histone and look at other things bound to it. So these are complexes, proteins. You have and I don't know the details, but if you can purify for histone 1 and then run that sample out on a gel and look at other things that are there. They have the hypothesis that CDK2 activity is inhibited by repression of E6 or E7. In other words, this. So P53 tumor suppressor activates P21, P21 inactivates CDK2. So the tumor suppressor pathway is on. CDK2 should not be phosphorylating histone H1. You can see over here in our HeLa cells, this is an antibody to phosphorylated histone 1, so unphosphorylated won't light up in the experiment. There's a lot in the normal HeLa state. CDK2 has 2 phosphorylation states. The bulk of it is not phosphorylated, thus inactive. But there's this band down below is the phospho form. It is active. And then cyclin E is a control. It's not going to change depending upon CDK2's activity. So cyclin E and CDK2 form a complex that's independent of whether CDK2 is phosphorylated or not. So, same thing now, we're infecting with E2, and in HeLa cells, infection with E2 shuts down E6, turns on 53, turns on P21, turns CDK2 off. So we lose that lower band. Histone is not phosphorylated. So we're stopping cancer by expressing this E2. In the E6, we're exogenously providing E6 and I haven't. Sorry, I still haven't drawn a chart in there. Sorry, I haven't drawn one pathway in there. So E6 is still present, CDK2 is still shut back off, loss of histone. When E7 is present, we don't lose the inactive form, we retain phosphorylation. So this is cyclin A also, that's what I was missing, inactivates CDK2. So either it's E6 in this case is not enough to inactivate CDK2. Cyclin A, because of P105 or PRB suppression, the tumor suppressor pathway, PRB, keeps cyclin CDK2 active.
So when CDK2 is inactive, it activates the histone, or the other way around.
CDK2 phosphorylates the histone, and that leads to proliferation.
So you're saying you need essentially you need the PRB to inactivate the CD15 in conjunction?
Yeah, that's what the data would suggest, yes. Another enzyme activity associated with proliferation is telomerase. So the telomeres are the ends of your chromosome that are produced by DNA polymerase. It's template-independent telomerase. When it's on, cells can proliferate. When it's off, cells become senescent because the telomere is short with cell divisions. What we have is an assay here for telomerase. We're looking at a target, and then when telomerase is active, it makes that target larger. And so we see an activity, telomerase is active, and then there's a time course here. These are days post-infection with E2. At day 2, on the control strain, we see some telomerase activity largely gone by day 6. In the case of E6 or E7, we see a persistence of telomerase activity post-infection with E2. So either one of them seems to be sufficient for continued upregulation of telomerase. Here, this is what I remembered when I thought I'll pull this paper for the class. So these are HeLa cells proliferating in a tissue culture dish. This is what fast-growing proliferating cells look like. They're clumps. You can almost see a tumor there. Cells growing very quickly. And then this is what senescent cells look like. They have a very different morphology. They're elongated. They have contact inhibition, so they don't grow on top of one another. Your cells and tissues communicate with one another so that they don't grow over. And so taking the control strain, you don't give it a virus with E2, that's the proliferative state right there. Provide E2, and I think this is taken at 10 days after infection, you've got perfectly senescent cells. So there's cured cancer in a dish with the expression of one polypeptide. With E6, exogenously provided, but shutting down E7, that's sufficient to cause senescence. E7 remains proliferative, except it's kind of halfway. I'm sorry. These are sort of senescent. You can see there are some extended morphologies in the slide. If you look at DNA synthesis percentage of the mock transfected cells, so 100 is how much DNA there is there, and then we're going to look at the same sample. If they're senescent, they're not replicating, they're not synthesizing DNA, so this senescence matches up to these two clones. It's effectively very little DNA synthesis because they're in senescence. The E6, almost as senescent, more DNA, but significantly less. And remember that this is sort of kind of halfway senescent, halfway proliferative, so is the DNA synthesis, as you would expect. So these figures are entirely consistent with one another. And so clearly the authors wanted to tease out why that was, or whether it was true, I suppose. looked at flow cytometry. We have this cool technique. We now have two instruments here at KSU. We have a flow cytometer and we have a FACS, which you could use in your Berlitz Carbon Summer Scholar Research Project if you play your cards right. FACS is sort of a higher tech, it's fluorescence assisted cell sorting. This is a technique where in a microfluidic environment, so a very narrow channel, cells are flowed. So what you do is take your HeLa cells, scrape them up off the tissue culture disc, get them in suspension, treat them with a protease, and do some other things, so that they're in suspension, and then you flow them through this microfluidic chamber, shooting them with a laser. And then there's a fluorophore in there. You can either use a fluorescently labeled antibody, or you can have GFP or something expressed as a condition of whatever you're looking for. And so that fluorophore being shot with the laser gets excited and then fluoresces. And the cool thing about flow cytometry is you can look at more than one wavelength at a time. So you can look at however many lasers you got and fluorescent dyes you have. You can look at different states in living cells. And the flow cytometer just measures them. The fluorescence assisted cell sorter is really cool because it has a little gate past the chamber, and if you say if something is X red, it goes in one tube, and if it's not, it goes in another tube. So we're looking at flow cytometry here based upon senescence-associated beta-galactosidase. So it was just observed, I don't know, 10, 15 years ago, that senescent cells express a beta-galactosidase activity. Beta-galactosidase cleaves galactose into monosaccharides for further metabolism. It's important here because there's a fluorescent substrate where if the beta-galactosidase is active, it gets cleaved and then starts to fluoresce. So what we're looking at here are degree of fluorescence versus number of cells in a flow cytometry experiment. So maybe they passed 20,000 cells past the laser and then looked at whether they were fluorescent or not. And so in the control strain, we have the mock are not fluorescent, and then transfected with E2 or infected with E2, they become fluorescent. So this is the marker of proliferating senescent. So there's proliferating senescent. These cells are fluorescent with this beta-galactosidase fluorescent substrate. These cells don't express beta-galactosidase, so they're not fluorescent. And then if we look at 16E6, remember this is the one that went proliferative fluorescent. This is only a couple of days. 10 days, yeah, sorry. So this is done at 6, this is done at 10. That's actually an important point to be. And then here is the 17, or E7 strain, and they're sort of kind of halfway. They're not as fluorescent, they're not as many that are fluorescent. So they're not as senescent. And indeed, you get, there's the mock infected, they're the infected, they're not this, but they're not this. They're kind of sort of halfway. This is a problem in a culture disc because what do senescent cells do?
They don't divide.
Right? They're not dividing. So those cells that are dividing overgrow. So at 10 days, there are more of them than there were at 6. So the authors were also interested in apoptosis. Or programmed cell death with the hypothesis that repression of E6 or E7 by E2 leads to apoptosis. And indeed that's true only in the E7 cells. So these are our fluorescence microscopy images of a staining for the nucleus. So this is just showing you where in this micrograph the cells are. And then the same samples, I believe this is the one that's 2 days, 5 days after infection. We're now looking at tunnel. So this is a dye that stains for DNA breaks, which is a hallmark of apoptosis. How death occurs is by breaking DNA. And we see that in the constitutive presence of E7 but not E6, apoptosis occurs. They looked at that also by flow cytometry. This is a more complicated flow cytometry experiment because we're looking at two fluorophores simultaneously. There's annexin 5 on this axis and propydium iodide on this axis. So in this experiment, proliferating cells don't take up either state. Apoptotic cells take up annexin. So they're not dead yet, they're undergoing apoptosis. Dead take up both. Propydium iodide is a membrane impermeable fluorophore, and so it's not going to cross into a living cell, it's going to go into a dead cell. And so with the mock transfected or mock infected cells, we get cells that are both low in propydium iodide and annexin 4. So each one of those dots is a cell flowing past the detector and either fluorescing with annexin or propydium iodide, both or neither. This is neither. These are dead cells, so there are some dead cells in the prep. When E2 is present, we still see little uptake of the dye. It's present in this quadrant. In the case of E6, we see the same sort of transition, a little bit more fluorescent, but not highly fluorescent. Essentially these cells, not fluorescent at all. And then with annexin, with the E7, we see a shift from not fluorescent over into the apoptotic quadrant. And so this result in microscopy is confirmed among the population that not everything is in apoptosis, but that we're driving the apoptotic pathway. Further underscored by a DNA synthesis experiment. So BRDU is a nucleotide analog. It is fluorescent. So when DNA gets BRDU incorporated into it, it becomes fluorescent. It is a marker of S phase. And so what we expect of proliferating cells to do have more DNA and to have new DNA. And that's what we see here. The mock infected cells, in the case of the HeLa cells themselves, go through, so incorporate more DNA, or incorporate the dye and have more DNA. So what you're seeing here is those are proliferating cells. The same is true of E6 and E7, and E2 is not supplied here. When we infect with E2 in the lower 2/3 of this figure, and we see a loss of the proliferative band here. So we're repressing E6 and E7. This is essentially a marker for senescence. There's some cells there. two copies, some cells that have one, they haven't divided obviously, but they've gone into senescence here. So lack of stuff here is an indicator of senescence. And then at 6 days, we see this, essentially the same thing in the E6 strain, and that leakiness in E7. So much more, many more cells down here in the non-proliferative state, but in the E7, there's some that are proliferating here. Over time, it's more of the population. So more here than here. So that taken together, this figure and this figure suggests that cells escaping senescence undergo apoptosis. So we have the apoptotic marker in the cells that are undergoing proliferation. As a result, or this, again, this is the hypothesis not shown in this paper, but that in, or I've erased it, in the mock infected cells, we don't have an apoptotic marker. In the E7 cells, we do. So the hypothesis is that you've got unbalanced proliferation or expression of oncogenes, and that lack of balance is what causes apoptosis to occur. And that may be why HPV has just has an E6 and an E7. Think like a virus for a minute. You don't want your host to undergo apoptosis. And so if you're going to suppress one tumor suppressor pathway, that's going to lead the cell into apoptosis. If you suppress both tumor suppressor pathways, then you get proliferation. Proliferation as a virus is good for you. It proliferates along with the host. So the question I didn't ask in the beginning would have been fun, which is why do you need E6 and E7? Well, the answer is because you do. But here's the data. You'll be, so I think I told you the story. I remember this paper because I once tried to get a job in this lab and I didn't get a job. It all worked out well. I remember Reno. But, so the guy, Dan DeMeo, you'll be happy to learn, wrote a paper despairing that virology was a dead science because it's all been done and they're bored. Everybody's bored by it. Fortunately or not, depending on your viewpoint, he answers, asks the question, is virology dead? And he says, no, it's not. For a number of reasons. It's a little opinion paper that I have posted under the Lagniappe section if you want to read about why people think virology is a boring subject. You didn't know yourself. But it's not. Notes. That's it.